Thank you for inviting me, and thank you for listening to it. So Bone Therapeutics is dealing with minimally invasive bone cell technologies, dealing with unserved markets, with a large pipeline and a strong IP portfolio, and in a cash position that enables us to go to the next major milestone. We've been founded more or less 10 years ago, and we have we are a spin-off of the Brussels University, and we have differentiated bone forming cells, osteoblasts, derived from MSCs from bone marrow, both autologous and allogenic. The allogenic gives us an off-the-shelf, able to deal with a large market. We have ongoing clinical validation, and I'll come back to that in terms of some recent clinical data, and we're working very hard on commercial readiness. The pipeline is based on 3 indications, 2 products. 2 allogenic indications with spinal fusion and delayed union. So in other words, what we aim here is from a bone marrow donation from a healthy donor, we obtain a product that is able to serve multiple patients. And we do that to produce bone-fusing vertebral bodies in patients with colon vertebral instability, or to repair a delayed union, a fracture that is taking longer to heal. Now, this has been what we're currently doing as an evolution of our previous product, Prehab, which is exactly the same idea in terms of manufacturing, bone marrow, differentiation, osteoblasts, ready to form bone, but the source, instead of having a donor, a healthy donor, is the patient himself, so patient to patient or donor to patient. That Prehab is targeting an orphan indication, osteonecrosis of the hip, and it's actually the one that we have now a partnership in Japan. Now let's go to the indications one by one. And shall I point to you that I'm talking about an allogenic product, in other words, donor to multiple patients. It's a large market of both in the US and in Europe. It's increasing, and it's a demanding market with challenging procedures and failure. I mean, the idea really is to avoid the back pain and the leg pain and the issues of bone instability by fusing the 2 vertebrae together. And the cells are there to promote that fusion. We recently press released a halfway interim analysis on our ongoing spinal fusion study, where we've seen initial interesting positive results. And should I point out that we look at the results at 12 months, where the majority of the studies looks at the studies at 24 months, because the idea is to actually produce bone fusion, but also produce it faster and of stronger quality. What you see there is the assessment in terms of dynamic X-rays. In other words, the inhibition of mobility, after all, that's what we want, is to actually stabilize the column, as well as actually bone formation. So in other words, the stability is driven by the bone fusion between the vertebrae. And what we saw is that in all patients we had a reduced mobility, and in 9 out of 15 patients in the 16-patient analysis, and we actually lost 1 patient that was actually not treated, we saw actual bone formation, continuous bone bridge between the 2 vertebrae in 9 out of 15. I mean, it's important. The regulators want to see mechanism of action, in other words, the radiology, but actually what is relevant is the functionality and the symptomatic improvement of the patient. And what we saw as well is a reduction of the leg pain, which is one of the major symptoms impacting the patient, and an improvement of the functional disability. You see it going down because it's a score, the Worcester Disability Index, which actually starts from severe disability coming down to just below 20, which is very slight to moderate disability. So a clinical significant improvement. So where we go from here, now this study continues to 32 patients. It is, as I said, an open-label, uncontrolled study, and we expect the recruitment very shortly, an analysis towards the end of next year, and a readiness to go to the next clinical studies after that. Now let's move to the other undeserved market, long bone delayed fractures. A certain set of patients, the majority of the individuals after a fracture have the healing of their fracture in about 3 months. If it goes beyond that, then the fracture is considered a delayed union. If it goes actually beyond 7 to 9 months, it's actually a non-union. So we're looking at improving the healing of the bone for that bracket of time frame between 3 to 7 months. It actually happens in a series of patients with, bad underlying conditions. And it's a significant large number of about 1 million, and it's increasing. And there's currently no opportunity for these patients. They wait and see. And eventually, if it doesn't resolve, then it's a major surgery with bone reconstruction. We actually approach this, and you can see there one example, and I'll come back to the detail. But just to tell you, the way we approach the treatment is via needle infusion of the cells at the site of a fracture. And so doing that with minimal invasion procedure. And we have conducted a study, again, open label, non-controlled, aiming at 32 patients with an interim analysis at 16 patients with a predefined endpoint that would lead to an anticipated stop in case the data would be positive. And that there was a target of achieving at least a 2-point improvement score in 4-quadrant measurement of the bone. And indeed, we not only achieved that, but we actually doubled by having an average 4-point increase in the 16-patient population. And you can see here, we go from, actually you can't see it, but anyway, you go from a fracture to a callus and actually a removal of external fixation. So this was actually associated again with clinical symptoms in terms of improvement of general health status and reduction of the pain of the fracture site. You see a bip there as the patient increases the mobility actually and is able to really go back to his life and with a consequent slight impact on the pain. So overall, the tolerability in this study, as well as in the previous study, was very good. We saw no significant side effects to report. We had, as in a lot of allogenic studies, a certain degree of immunogenicity, and we had one event that was not related to any other apparent cause, so we cannot exclude a potential relationship with ALOB. Overall, this study, Overall, the study was so positive that the DSMB, again, with predefined criteria, suggested that we would stop for efficacy, which enables us now, as we finish the analysis of the patients, to go into the next study, which will then be a 2B controlled study planned for 2018. A third indication now with pre-op, the autologous, is for osteonecrosis of the hip, an orphan indication, again with no standard treatment other than decompression or bone marrow concentrate introduction in the osteonecrotic hip. As you can see, we have already presented a controlled study versus bone marrow concentrate, where we showed overall significant positive response measured by absence of fracture and pain improvement. both at 24 and 36 months. So clearly showing that the cells, what they do is they prevent fracture by inducing bone formation with a consequence clinical of reduction of pain. This study, this indication is currently in a phase 3 with a predefined prospective planned interim analysis to be done towards the second quarter of next year, which will then enable us to be able to go to the regulatory discussions after that. Now, this product is now in partnership with us, like I say. Again, and as I mentioned before this morning, it's part of the interest in Japan. It's an orthopedic expert company that wants to go into cell therapy in view of the developments in Japan. So clearly representing an opportunity to go there, and clearly they will take it and develop it further. So we're actually very much also working on optimizing the product through cryopreservation, increasing the yield, and scaling up the automation. It is crucial to devote aspects in terms of process development and have your product ready for large-scale manufacturing and commercial scale. Now, obviously, like all of these companies, we have a runway that is limited, and where it will be no surprise to you that in view of that, we're planning to go again to the market pretty soon. And we believe that the idea to have documented clinical data that we want to confirm in a controlled study for these indications with a product like this, it's a valid proposition which will be attractive to the market. Time will tell, and we'll see. What we're going to achieve in 2018, we're going to complete the spinal fusion study. We will have the final results of the delayed union. We will have this interim analysis of osteonecrosis study, which will enable us to conclude on that indication and engage on potential regulatory discussions. And we will go into the next phase to be a controlled study for delayed union. We're also very much working on the product optimization, and we will continue to engage with our partner now. And in 2017, we delivered against the expectations, and I've just went through those expectations and those deliverables to you in the previous slides. So it is an off-the-shelf allogenic platform. It goes for a large, significant market, which doesn't really have valuable options. It is going through clinical validation with preliminary results, and we're working hard on commercial readiness to make sure that the overall business model and the value proposition it's good for all key stakeholders. Thank you. Any questions? Majority of them are young adults. I mean, the fracture is really for active, as was a skier there. Osteonecrosis is young, middle-aged adults. Spinal fusion, it's probably a little bit, older, but all of them in active ages.
Thank you. Thanks, Miguel, for the talk. Do you guys know yet if your mechanism of action is cells interacting, or if it's trophic factors and causing cells?
Yeah, good question. It's both, actually. In animal models, we see the induction, using human cells in an animal, we see induction of the animal own bone, and we see engraftment and persistence for a certain time of human bone based by the cells. So it's actually both. It's engraftment and induction. All right? Nobody wants the 20 last seconds? Done. Okay. Thank you.